Hello everyone, my name is Nattagh and I'm going to be presenting my work on gold nanoparticles and their applications in radiation cancer therapy. Specifically, I want to talk about how gold nanoparticles can help enhance the damaging effects of radiation. And if they do, I want to see if this enhancement is due to a change in the number of DNA double-strand breaks happening in the cells that are irradiated. Before I talk any further about my work, I want to go over some fundamentals of radiation therapy. In radiation therapy, you often have a tumor surrounded by soft tissue, and you're trying to deliver a lethal dose of radiation to that tumor. Clearly, the radiation will have to move through some healthy soft tissue before hitting the tumor, so there's going to be some collateral damage. This collateral damage can be visualized by this cell here. Excuse me, by this graph here. If you want to achieve 100% toxicity in the tumor, you're going to probably want to deliver a dose of radiation that'll give you a lot of healthy cell damage as well. So that's a problem. And over the last decade, the last century that radiation therapy has been used in the clinic, there have been a lot of improvements technologically and theoretically. One of these improvements is the use of nanoparticles in radiation therapy. And that's what this presentation is about. Specifically, gold. This is the very basic setup of how it works. Suppose you have two hypothetical tumors. Same size, same everything. One tumor is just a regular tumor, and the other has gold nanoparticles distributed throughout it. If you hit both these tumors with the same type of radiation, same energy, you'll get a different result. And the tumor that had gold will shrink more, will get more damage. You might be wondering why this is, so I'm going to talk about the physics. And I know this is the end of the day and we're all tired, but I promise you this is the only equation we'll talk about. The physics behind this effect is basically the photoelectric effect. And in the photoelectric effect, you have a photon hitting an atom, the photon gets absorbed by the atom, and an electron pops off. Now, every time a photon interacts with an atom, the photoelectric effect does not occur. It occurs with a certain probability. And this probability is proportional to the cube of the atomic number of the atom being hit, and 1 over the cube of the energy of the photon doing the hitting. So, if a photon hits an atom with high atomic number, an atom such as gold, as opposed to water or soft tissue, you're going to get a lot more photoelectric effect. And thus, you're going to have a lot more electrons. So, the good thing about electrons is that they have short range. And what does this mean in the clinic? This means that when a photon hits a tumor, it does some damage and it moves out of the tumor. It leaves the tumor because it has a long range. But in the gold nanoparticle case, the photon comes in, most of them turn into electrons, and then the electrons, since they have short range, they stop in the tumor. They basically bounce around, do a lot of damage, and all the energy is deposited right there in the tumor. It doesn't pass through like a photon does. So this is good. This means you can damage more of the tumor that way. And the way we set up the experiment is like this. We got 4 dishes. We plated the same number of cells in each dish. Our control was just the regular cells. Another dish, we're exposed to a certain concentration of gold nanoparticles. Another dish was exposed to a certain dosage of radiation. And the last dish was exposed to the same concentration of gold nanoparticles and the same dosage of radiation that the other 2 were. After we let them sit like this for a while, we counted the cells that were left over. And we plotted our data. On the y-axis, we plotted the number of cells, and on the x, the concentration of the gold. On the left, the dish is the one that was irradiated, and on the right is the one that was irradiated and exposed to gold nanoparticles. Clearly, we see a decrease in the number of live cells when we had gold with the radiation. You might think, okay, great, now we got radiosensitization. But this isn't necessarily the case, because who's to say that the decrease in the number of cells isn't due to the toxicity of the gold itself? So we plotted some more data points. On the top, we have our control, and we see there's the most number of cells there, which makes sense. Here we have the ones that were just exposed to gold. And the difference between the number of cells here can be attributed to the toxicity of the gold. So, the greater distance between these two plates, as opposed to these two, makes us strongly suspect that there was radiosensitization that occurred here in the bottom right dish. To make it more clear, the slope is what needs to change for radiosensitization to occur. The slope of the top line is attributed to the toxicity. And if the slope of the bottom line is the same, we can tell that the gold didn't do anything, didn't synergize with the radiation to do more damage. But obviously, we see that the slope is steeper. So once we knew that we could get the radiosensitization, we wanted to see why it was happening biologically. It's commonly accepted that radiation does damage to cells via DNA double-strand breaks. So we looked for a way to quantify these DNA double-strand breaks. And it was nice to know that there are proteins that accumulate at the double-strand breaks, and they're meant to repair them. Fortunately, we found a way to attach antibodies to these proteins, and these antibodies are fluorescent. So, since we have fluorescent antibodies that attach to the proteins that accumulate at the DNA double-strand breaks, we can effectively get fluorescence at the location of the DNA double-strand breaks. And then, if we count all these bright spots that we get from fluorescence, we can quantify our number of double-strand breaks. Here's an example of what I mean. In the dark blue is the nucleus, and the bright green spots are the fluorescent, or accumulations of the fluorescent antibodies. So, after we got images of the bright spots in our nuclei, we sent it through a computer algorithm that counted the number of bright spots throughout thousands of cells, and we took statistical averages of our data. The setup was basically the same as the last one, except for the last step. Instead of counting the cells in the dishes, we attached the fluorescent antibodies and imaged. This is an image of my cells, and we're currently in the phase where we're trying to send it into the algorithm to get the results from the number of bright spots we have, so we can compare it with the other dishes. So, our expected results, once we get the data back in the algorithm, will be something like this. This is somewhat the inverse of the last graph, and instead of having the number of cells on the y-axis, we have the number of bright spots. Please also keep in mind that on the last graph, the irradiated dishes were on the bottom and now they're on the top. And just like the last graph, the slope is going to be the determinant. Another step we're going to take soon is we're going to use new equipment. Since this research is meant for the clinic, we thought we should use clinical devices to irradiate our cells. In the lab, we use smaller laboratory-grade devices that can produce kilovoltage X-rays. But in the clinic, they use much stronger devices that can produce megavoltage X-rays. So, we want to repeat all our experiments with a clinically relevant machine. In summary, we saw that gold can sensitize the cells. And we believe that this sensitization is due to an increase in DNA double-strand breaks. And that about summarizes my project, but I would like to talk a minute about what the future of gold nanoparticle technology, what it is, where it's going. Because we eventually want to get it to the clinic, and currently it's in the animal studies phase. I thought this picture was really cool because it represents the major problem that this technology is having before it can move to clinical trials. And that problem is delivery. This is an X-ray of a mouse's head, before on the left and after on the right, gold nanoparticle injection into the tail vein. The bright spot is due to an accumulation of gold nanoparticles in a brain tumor. You might wonder why, if you injected gold nanoparticles into the tail vein, why they accumulated in the tumor here. Well, that's due to a natural mechanism of the body, and that is that tumors have poor quality vasculature. Vasculature is leaky. So this gold will just pass through the permeable vasculature of the tumor and disperse itself around the tumor. Healthy tissue is not as permeable. But the problem is that the gold deposits in other places as well. Places such as the kidney, the liver, and in this case, you can see there's some other spots in the mouse's head that have gold. And this is the main problem. This is what scientists need to solve before it can move to clinical trials. And for example, one of the techniques they're using is they coat the gold nanoparticles with some organic molecules that can help get these molecules to target the tumor and only the tumor. So hopefully, if we see that happen one day, this technology can move to clinical trials and eventually clinical. Thank you for listening. I'd also like to say thank you to my lab partner, Lucy Chow, my PI, Salon Costas. Also thank you to Surf Rose Hills, Leia, Carol, Nathan, and Eva for all your hard work. And I'd love to hear some of your questions.